Okay, maybe
Then
you
maybe we
can
can
start.
maybe
Yes.
we can start now. John, you ready?
Yes, go
Okay.
ahead.
hmm So good afternoon, everyone. I'm very happy, extremely happy today to have Professor John Donoghue with us. We've been looking forward to John's talk for a while now, and I'm very happy that we are having it today. And I would like to welcome you all warmly to this talk entitled Two Decades of Brain Gate, Reminding Mind to Recreate Movement and Communication. Let me give you a background about John. Well, I would presume that most of you know Professor John Donoghue. Professor John Donoghue is the H.M. Briston Professor of Neuroscience and Engineering at the Brown University. He's known for translational research in human brain-computer interfaces to restore movement for people with paralysis, known as BrainGate, as well as for fundamental research to explain how networks of cortical neurons compute actions from plants. At Brown, he was the founding chair of the Brown Department of Neuroscience, the founding director of Brown Institute of Brain Science, now called the Carney Institute, and the Department of Veterans Affairs Center of Excellence in Neurorestoration and Neurotechnology. Donoghue, Donoghue was a member of U.S. Brain Initiative, first NIH Working Group, and is a fellow of National Academy of Medicine, American Academy of Arts and Sciences, as well as several other academies. His award includes the German Sulz Prize in 2007, the Roche Nature Medicine Prize in 2010, the Schrödinger Prize, Germany in 2012, and the first Israeli Brain Technology Prize in 2013. And without further ado, I would like to invite Professor John Donoghue to give his talk to us. Thank you so much, John.
Oh, thank you, Robin, and thank you for the opportunity to talk today. Excuse me. I just want to say that it's been a pleasure to get to know everybody that's affiliated and working on B-CRATOS. It's an exciting project, and I'm participating as an advisor, and it's great. Not only a great team, of talented people, but a very important project with a lot of great innovations that are really exciting for the whole field. And so what I'm going to emphasize today is discussing brain-computer interfaces, and particularly the work that's gone on in my lab for this project we call BrainGate, and I'll talk more about that. But just to start, this is Matt Nagel. It's 20 years ago, actually 20 years ago just now in June. And Matt was injured from a knife injury. He's completely paralyzed from the neck down due to a spinal cord transection. And Matt was the first person to have this electrode array implanted in his motor cortex and to use a computer to try to see what we could do with a brain-computer interface to restore the ability to control devices or the body with these kinds of technologies. And so there's Matt's, you can see sitting in his wheelchair. He's got a respirator helping him breathe. There's a technician overseeing the whole operation. And the important thing is that he's getting to play a video game on the right-hand side. And he can, excuse me, he can control the cursor to erase those little treasure chests and hopefully avoid the goblins that are the little black squares. So what I want to talk about is the sort of history of what's gone on, particularly in my lab, but mentioning a bit along the way of the important other work that has gone on. In this, it's really generally part of something that I think of as neuroelectronic medicine. It's an emerging field developing devices for brain therapy, for brain repair, brain restoration. And when I use the word brain, I'm using it as a synecdoche, which is a word that I just learned in English, which just means it's the part of something that means the whole. So I mean the whole nervous system, but I will use the word brain here and again. And specifically, I want to give a few historical highlights, and I mean just a smattering of key things that are kind of fun and interesting to know about, simply to show you how ideas evolve, and they certainly don't happen overnight. They actually, this brain-computer interfaces and neuroelectronic medicine is something that we can say has been evolving over thousands of years. Then I want to talk about the 20 years of BrainGate in humans, and then I'll end by talking a little bit about the future of neuroelectronic medicines in general. So just for this touch of history, Scribonius Largus was using electric fish therapy, putting it on the heads of, including the emperor at the time, to treat migraine. It was a kind of electroanalgesia. And in those days, of course, they didn't really know much about electricity, and they certainly didn't know much about the brain. But it was how many things evolve. There's actually a use of something from just practical observation without really understanding anything about it. Jump ahead almost 1900 years, and thanks to the work of Galvani, we understood this relationship between the special electrical nature of the nervous system and the body and the brain. And his colleague Aldini and others were out in the world in those days after they had learned that electricity, which was still a sort of a new thing to play around with in those days, they could take a cadaver, a freshly deceased person, and electrically shock the body, and they would jump. And this was almost like a grifter show that was frightening people, to try to understand how could this magical power reanimate the body. In fact, that was even an inspiration for Mary Shelley to write Frankenstein. But the more serious advances, I think, with the 1930s and 40s especially, but maybe the 50 years before that, and certainly now the 50 years after. And I think two of the really significant advances are involving both the tools, and then these tools enabled us to understand the mechanisms of neural function. And Edgar Adrian was really a giant in this whole thing, where he used microelectrodes, fine wire sensors, and what recently developed vacuum tube amplifiers to take small electrical signals and make them larger. And his major discovery of several was that neurons fired action potentials, these spikes, these single impulses, and these impulses were the code. And so he said, the message consists merely of a series of impulses. And he won the Nobel Prize for that in 1928. Then the other significant event at that same time was that electrodes put on the scalp, these were more macro electrodes put on the scalp and amplified, could show more macro scale brain activity. And Hans Berger and a few others even before him were sensing these patterns of activity on the brain. And here what you could do is measure things like brain state, like different stages of sleep or alertness or inattentiveness. So these were major milestones in discovering, that there was electrical activity in the nervous system, and that it actually meant something, and that we had the tools that were necessary to sort of assess these signals. And certainly in the 19th and 18th, 19th century, there was a big fascination with these therapies, and there were kinds of neuroelectronic medicines with all kinds of claims, like this one, that you could treat all kinds of diseases and cure them, cure paralysis, lost vitality with electricity. Even Benjamin Franklin, if you follow U.S. history, was an extraordinary inventor, was playing around with these kind of devices. But today we really have come up with a more mature thinking and approach towards trying to apply our understanding of the electrical properties of the nervous system and use it as a neuroelectronic medicine. So these are some, I think, the way that therapies are organized now, and this may not be permanent, of what you can do now or what you might be able to do. So basically, you can stimulate, you can record or read out activity, or you can use them both together in what's called a closed loop system. And for all engineers, that's a well-known use of inputs and outputs. So under stimulation, you can modulate, and that's usually done with a macro kind of larger stimulus, where you can use these stimulation to reshape aberrant brain circuits. That's been successfully applied to treat Parkinson's disease and seizures, and a little bit and now there's ongoing research work to treat depression. It's also can be perhaps used to regulate even metabolic activity to slow neurodegeneration. So that's another application of these kinds of macro level stimulations. Then more specifically, you can use it to write in detailed information to restore sight or hearing or touch. And of course, in hearing injecting electrical signals into the cochlea to treat deafness is really an extraordinarily successful restorative use of stimulation. Then to record, that also can be global or specific. When we globally look at brain activity, like Berger did, we can look at the state of the brain. There are efforts to look at changes in brain mood, like depression or anxiety, states of sleep, and even to detect seizures as a diagnostic, which is a well-known and long-standing use of recording in the EEG. And then we can also use it to record intentions. That is the detailed thoughts going on in your head, specifically, as I'll talk about in much more detail for BCIs or brain-computer interfaces, to create movement commands and then make devices to restore movement. And as I mentioned, you can use them in closed loops where you can read out the system, make some decision then to intervene to treat a disorder. And these devices, of course, come into what are called, wearable or implantable devices, sometimes called invasive or non-invasive, depending on whether you cut the skin or not to use the sensor or the system. In general, the wearable systems are currently lower resolution as a general statement, and the implantables are higher resolution. But that may not always be true. But I think that I like this designation of things that are on the outside, things that are on the inside as a wearable and implantable designation. So specifically, I'm going to talk about now the use of readout to make a brain-computer interface. The project we've called for the past 20 years BrainGate. And this BrainGate started out in my lab as a lab project to trying to understand how the brain is coding and transforming information, but has developed over the years into what we call the BrainGate Consortium, which consists of a number of universities that are working together. And I started that, but is now very ably led by Lee Hochberg, who leads this team with Stanford, Emory, MGH, UC Davis, and it may be an expanding group. And so I want to emphasize that one of the things that makes this a particularly challenging enterprise to develop all neuroelectronic medicines is it requires the knowledge of neuroscience, the tools of engineering, and the application in clinical medicine. And it requires really a special culture of interaction. It's not something that works when each of these are done independently. And so that's a very hard and I think a novel problem to actually create medical therapies by integrating such diverse fields with different cultures, different languages, different disciplines. But this is what's happening, and I think it's really an exciting time to bring all these fields together. And it seems to be working with even with organizations like BrainGate, and also B-CRATOS is another example of this kind of collective action and really a way to advance our understanding of the brain and to create the tools that are needed both as therapies and to understand the brain. So one other thing that's important, another fact that's an advance that we think of as rather recent, but is something that is been around a while, and that is that the key to understanding sort of what's going on in the brain is not something that you can study one neuron at a time, but actually is a pattern. It's really the patterns of activity that are spread across the brain. The little schematic on the left I'm showing is a bunch of flickering QR codes. And I use that as just a schematic way of demonstrating patterns of activity where maybe each little black dot is a neuron that's on, and then it turns white when it turns off. And so this is what's going on, say, whenever you're looking at something and deciding to move to pick up your water bottle or your coffee cup. And this is not a new idea, actually. Adrian, again, this giant of neurophysiology, was one of the first network neuroscientists, if not the first, who said that the key thing to understand is the interactions of large numbers of neurons. That is the sort of population or ensemble of neurons that are interacting. And in a more modern demonstration of that, Jack Gallant has got one beautiful demonstration using fMRI, which is slow, but it gives us a very global perspective. And the red and blue spots are areas of up and down activity in a brain while a person is pretending to drive a car in a video game. And the point is that there's a lot going on in the brain all over the place when you're doing something as ordinary but complex as a sensory motor behavior, where you use vision and transform that into action. So that says if you want to make a brain-computer interface like the one that Matt Nagel had, you need to know something, that you need to tackle understanding these patterns of activity, and you need to know where to go. The little red dots on the right-hand picture are all the sites where we know that there's information about, say, moving your arm. This is a summary diagram from John Kalaska. And but we do know from now actually more than 150 years of neuroscience that there's one kind of special spot that's a primary candidate, the primary motor cortex, for getting these patterns of movement activity, say in a person that's paralyzed. Although all of these others are candidates for getting movement signals, and many of them have been tried, and many of them are also sources. But all of the work we've done is focused on either primary motor cortex or these PM premotor areas that are nearby. So how do you make sense out of these codes? So I like to use the QR code as a simple way to give you an intuition of what we do, rather than go into the details of the various models that have been applied. So think of, again, each little pixel that represents a neuron in an on or an off state. And of course, in the brain, these are flickering at about 50 times per second. And so it can produce a complex set of thousands of patterns. And with something like the electrode array that we developed, it was something that Dick Norman originally developed this array, and then we took it into make it something suitable for humans. If you were to sample from that pattern, of course, you would get, in this case, 96 out of thousands of possible neurons. You could sample that pattern and ask what's going on. So the way we do it is actually rather simple. We ask, say, in a monkey implanted with this electrode array, move to the left with some cue, or move to the right. And with each of these movements, we just simply record what is the pattern of activity that occurs through our electrode array in the neurons. And then we store that pattern. And then when we detect that in the future, when we're using or decoding the output, we say, if we see that pattern again, we move a cursor, move the arm, move whatever the device is to the left. And we can apply that to all kinds of variables, and we can and apply different kinds of decoding models. But it's basically that is the approach. Is one pattern means a particular encoded variable. A lot of people do not understand that this doesn't require any learning by the user of a brain-computer interface. It's simply reading a pattern, understanding what it means by building an encoding model, and then decoding it. But that doesn't mean that learning is important. With learning, we can get a better sense of the code, and people can learn better to control cursors. I'll show you some of that work in a few minutes. There have been a whole range of models to make sense of these patterns of activity, and people are using many approaches, from pattern theory, geometry, dynamics in the engineering sense. And these have helped improve our ability to interpret limited samples from complex patterns as to what they might mean. And so I think we're making progress, but we don't know, we don't have a full understanding of these patterns. And I want to emphasize that, we did some of this work in our lab, particularly the real-time decoding of these of information. But there's a whole long list of people, starting with, in particular, Apostolos Georgopoulos, saying that you could, in fact, make sense out of populations of neurons in terms of real-world control variables. So just to show you that it actually works, I think this is an experiment that I love that was done in our lab by Carlos Vargas Irwin and computer scientist Michael Black. Excuse me. And what we did is we just had a monkey reach and grasp for objects that were swinging in front of him. He had a motion capture system that captured all of the arm movements. And we recorded the motor cortex while he did that. And we asked from that encoding model that while the hand was moving around in space, how well could we do at decoding not just one variable, but the entire motion of the arm from the shoulder down to the fingertips? And in fact, this reconstruction shows that we could do a pretty good job with a small sample of 96 channels from that little electrode array placed in just a part of this motor cortex, the arm area of the motor cortex. So in the movie, the ghosted image is actually what the monkey was doing to catch an object, and the solid is the reconstruction. And you can see we're definitely not perfect. The mismatch means we're not either off in time or space. Nevertheless, it's remarkable that we can decode from this limited sample the entire arm, and that's one of the, I think, important advances is small samples of the motor cortex, very randomly selected in that arm region, gives us complete information about aspects of finger, wrist, elbow, shoulder movements, good enough that we can reconstruct the whole action. So I think this was very encouraging that we could in fact do something like use all of this knowledge to make a brain-computer interface. And in about the late 1990s, when we had done some monkey work and looked at the work of others, and we said, well, we know where to go. We know we have a tool to record the activity. We can make sense out of it. Could we, in fact, recreate a new bridge between the brain and the outside world for people who have lost that connection, people with strokes or spinal cord injuries, or degenerative diseases like amyotrophic lateral sclerosis? And in fact, so we did that. We said we have this neural interface. We can read out signal patterns. We have this decoding schema, or one of many. And we said if we could make a useful command signal, we could do things like reanimate the body that's paralyzed, restore speech to people who couldn't speak. We could also connect these command signals to computers to get them to do all the things they do, to robotic arms, and we haven't done this, but others have, to connect these signals to exoskeletons to restore mobility. So just to say, give you a hint of people that who could benefit. There's something on the order of 130,000 people every year that become paralyzed to some degree, and have really no other major disorders like cognitive or other kinds of sensory loss. And you can see the extremes of these two here on the left. This is not a still picture. That's a movie of a young woman who's completely locked in from ALS. She can't move anything, can't blink her eyes, can't speak, so she has no means of communication at all. And on the right-hand side is sort of the other end of the spectrum, as a man with has had a stroke, and he's got limited or degraded movement on one side of his body, of his arm and his leg. And you can see how he walks as a consequence, and he can't use his hand in very productive ways. So then there's the full spectrum of these disorders, from ALS and stroke and spinal cord injury and even traumatic brain injury and other disorders. So there is really a big clinical need where you could help people, where the need for a person who's locked in, totally paralyzed, communication to say, I'm in pain, I need medication, I have a discomfort in my foot or something, would be major. On the other hand, the kind of devices you would need for someone with hemiparesis would be, say, in their case, they want to do more complex tasks using their paralyzed hand. So in the late 1990s, we had this vision. And so the idea was that someday we would create a device that would make a person paralyzed look like anybody else. They would be indistinguishable from anyone. We would put this array of electrodes and sample their motor intentions. Through an implanted device, we would process these signals and somehow radio them to the outside. When they got to the outside, we would then put them in a computer that's in a smartphone, be able to process those signals, then transmit them to an electrical stimulator, which could be connected to the arm, and it would either stimulate the nerves or the muscles and reanimate the arm, so that when this person had that device properly implanted and placed, they would be able to do all the things that you and I take for granted every day, and it would be so good you wouldn't know the difference. Well, that's a vision. That's not something that we can really do now yet, but you'll see what the progress we've made. So I showed you already Matt Nagel. Back in 2006, we published the results of the study with two patients, and the important findings is we showed that, in fact, even after long-standing paralysis, the motor cortex was still functional. That is, the neurons were active, and that was, one of the first major advances. We didn't know what would be going on in the motor cortex of a person who had lost all use of their arm for years because of an injury, for example. We also showed that the decoding works, that we could actually make sense of these signals so they weren't scrambled in some complex way. And this was a great validation that preclinical work in animals really translated very well to humans. And we also found out that Matt here is imagining moving his hand because he really can't do it, but it looked very much like something was really happening when we were recording those signals. And in this early days, in the first couple of years, around 2004, we showed you could achieve at least 2D control, which was good enough to operate a computer, here to play a video game, but to spell. And in fact, we showed that Matt could control devices. And if you should listen carefully to this, because this is Matt controlling a prosthetic hand on his lap. So it's controlled by his neural signals. All it does is open and close. And the importance of this demonstration is how he reacts to being able to control something. Remember, he's paralyzed and hasn't moved his hand in years. So it was really, a profound moment for him, and something that was liberating, but actually wasn't very functional. So in the next several years, we concentrated on trying to make better control. Remember, Matt just simply moved the cursor on the screen. We showed that we could improve the quality of the cursor movement, and also we could achieve a click by thinking about squeezing the hand. So we could get point and click movements, which is how you can operate a computer. Shows here two examples of using that in what's called the center out task, going from a dot in the middle to a target on any of 8 directions, which again, as I said, was predicted by earlier work in monkeys, starting with Apostolos Georgopoulos' work. And then shortly thereafter, we showed that, in fact, you could use these signals to get multidimensional control. And here is a famous video from Kathy Hutchinson showing how she could, in fact, control the motion of a hand in space, and even pick up a bottle that had her morning coffee in it, and bring it to her face, and be able to operate it well enough to take a drink on her own. So she was unable, due to a brainstem stroke, to be able to do anything with her hands that was useful. But here she can actually manipulate that her and drink on her own. So you could control cursors, computers, robotic limbs. And in fact, in 2017, we made the step of showing that you could actually reanimate the body, in this case, the arm and the hand. And here's Bill, who has 2 arrays in his head. You can see that gray box on his head is the connector, and there's cables coming out, going off to instrumentation that decodes. But importantly, command signal is fed into a stimulator that's attached to various muscles in his arms. You can see the elaborate nature of that on the diagram on the left. But with this, he's able to think and open and close his hand and move his arms so that he can actually hold on to a cup and take a drink, as he's doing here. He was able to eat and do a number of other things with his arm as a consequence. So this is a lab level demonstration done with the Case Western folks, Bob Kirsch, Hunter Peckham, and Balu Ajaboy, who are really the masters behind these FES, or functional electrical stimulation systems. So we provided the brain signal, they provided the arm signal, and we brought the two together to show that, yes, it's possible to reanimate the body with this brain-computer interface design. Jamie Henderson and Krishna Shenoy, who I just want to mention very sadly passed away just a couple of years ago. And it was a tremendous loss of an absolutely brilliant and wonderful human being who made major contributions to this whole BrainGate group and to the field. And what they developed and showed is that not only can you, say, maybe type by pointing and clicking on a screen, that you can imagine handwriting and handwriting individual letters like those shown on the right. Those are the letters that a person was asked to think about, and these are the decoded letters that they made. And then we put that through a decoder, and you can see here's an example of copy typing in which we asked the patient to retype the image, the sentence above, and you can see that there's really quite rapid ability to create and decode the signals. And in fact, this was typing that got up in one case up to 90 characters a minute, which is sort of what you and I do. So we were able to get very quick decoding of imagined writing into speech, so you could write notes and be productive, moving towards communicating with a brain-computer interface. And about the same time, within a couple of years, Eddie Chang has done something similar, now using a surface grid to decode, and has had a successful 30 characters a minute kind of decoding of words. And this shows actually two approaches that are evolving. One is with this electrode array, it's high density sampling of precision signals, single neurons or groups of small groups of neurons, or a much larger spatial extent of the brain, but at a lower resolution recorded from the surface of the brain. Both of these seem to work, and both of them are being pursued as ways of being able to decode signals. And I think, right now we don't know, is one better than the other, or what are the advantages of one over the other, or maybe both of them will be productive avenues. So over the past 2 decades, the reality is, we and BrainGate got to the point of reconnecting the brain to the body as well as computers and robotic arms. But we haven't really reached the vision that I had. I naively thought in the early 2000s that within 5 or 6 years we would have this device on the right done. We're still far from it. In fact, I think something that initially disappointed me, but I think we can understand the complexity of this whole field, is that 18 years ago, our publication of Matt's success led to a front page story on the New York Times: paralyzed man uses thoughts to move a cursor. And then, if you remember, just recently in 2024, 18 years later, Psychology Today's headline for the Neuralink demonstration of their technology was "Paralyzed Man Uses Thoughts to Control a Computer." So in 20 years, we have just about the same headlines. And in fact, Neuralink reported that they have malfunctions in their technology. I'm not criticizing them. It's very important that we move forward to commercialize these devices to reach patients. I think this points out what a big technology challenge this is, how the interdisciplinary nature, the need for skills and infrastructure and resources from multiple fields is really a barrier that is something that's hard to overcome. And but I'm not as initially I was pretty upset that it's really taking us so long. I think we have some solutions, and I think we're at a point where we're really going to take off and do a lot of things. So I want to point out some of the things that are happening that are making our ability to make successful brain-computer interfaces even better. So a couple is that we have sort of smaller devices that can sample many more signals. The BlackRock array that we use that was developed by Dick Norman and me for humans is people are using multiple ones now, so you can get hundreds of channels, so we can sample more areas with high density. The Neuralink company has introduced these threads, these wires that go in. Each one of them has multiple sites, not just one site, like this multi-electrode array we were using or are using. And then so that can record hundreds of cells. And they also produced a implantable device that can process and transmit the signals to the outside, though not at very high bandwidth. So it's a lower resolution of spikes than we would like to have, but it still allows an implanted device to transmit hundreds of channels. And then, although as I pointed out now, because it's not working, they're getting in the 60s, I think, is what the local recent reports are. The V Center in Geneva, where I was for a while, is also developing a high resolution 128 channel system so that will amplify and broadcast to the outside. So this is an implantable system. And the Chang Group is also developing their 128 channel surface grid. So these are all advances in sensors. There's one more that's coming from Synchron, which is a very different approach to put a lower channel count device based on a stent into the brain vasculature, particularly the sagittal sinus. And that can sample, but it's a fairly low resolution EEG-like signal, but still is another approach and another way to get a sample of brain activity. But I really think what's coming is really going from electronics that I think today are more like, the first Hewlett-Packard mobile phones that were really bulky and heavy and not so sophisticated to something that is like the iPhone of today. It's got lots of electronics and lots of capabilities. So the one of the exciting ones I think that's here now and in heavy use in research is the NeuroPixel electrode. It has 960 channels per probe. So when you insert one probe, you get almost 1,000 channels. So if you were to put 10 of these in the brain, you would get 10,000 channels or more. So huge samples. And they have built into them the initial processing units. So it's reduced what used to be sort of a toaster size processing amplifier down to something that's the size of a fingernail. So there's real promise that we can get much better technology, and this is coming in the future. I'm very excited about this. So, you know, let's sort of summarize what the future of this high resolution neurosensing might be. So I think we already know that the potential for BCIs is tremendous in terms of restoration of function. Using a computer, it liberates people who are paralyzed. It can provide mobility, like Gregoire Courtine has been showing by sensing the spinal cord instead of the brain, but still a command signal to control the legs. It can be used for communication, as I showed you. And there's now beginning of ideas that we could use these BCIs to promote rehabilitation. That is, if you use a paralyzed arm, not only do you regain it with a BCI, but perhaps using the BCI may promote rehabilitation and restoration of function. But that's an avenue to explore. Um, I think that these same neural sensing systems, BCIs, can be used as diagnostics, for example, to predict seizures, to say alert a person that they might be going to have a seizure so they could take preventative measures. And then they could be used to measure where a person is that is presumably in a coma, but what is their level of consciousness? So we could use it as a diagnostic, as a measure of brain state. We can also use it in what I call neurofeedback, or it's called responsive stimulation. Can be used for epilepsy to pick up the fact that there's a seizure and try to stop the seizure. And NeuroPace is already a device that's out and available to patients and successfully helping to diminish epileptic seizures. But these same device closed loop systems could be used for mood disorders, memory disorders, and a whole bunch of other applications like that. And it also could be used as drug or disease biomarkers to look for patterns of change that are going on in the brain as a result of a disease or the result of a drug. And so you could predict whether a patient is responding to a drug, whether they have the emerging biomarkers for a particular disease. These may be more candidates for wearable technology than implantable technologies, but both, again, may be real valuable applications of this kind of neurosensing technology. So I think we're really at a point in the 20 years that since we first started with our brain-computer interface and with all the other advances in neuromodulation and cochlear implants and closed loop devices like NeuroPace and now all these future applications, that we are really making progress. But I still think there's a big challenge that going from concept to a real world device, we have this real valley of death gap between the concept and all the clinical neuroscience, computational, mathematical, statistics, and engineering barriers to get to a real world product. So I'm actually working to try to help build an institute that will fill this translational gap and provide in a nonprofit organization skills and infrastructure and resources that can help accelerate and overcome these complex challenges, bring together many disciplines, and create that special culture that I think will get us into producing more, better, and better kinds of devices and much faster. So all of this work over the past decades has involved just probably more than 100 or hundreds of people. And it includes people from my lab and people from the BrainGate group, as I pointed out, with Lee as one of the leaders. And so I can't go into naming everybody, but I want to acknowledge that this has been an enormous effort by large numbers of very talented and hardworking people, some of whom you've just recently heard from, like Misha Soroya. And I want to make sure I acknowledge and thank all of them for their making all of this possible and pushing the field forward. So with that, I'll say thank you for listening. And if you have any questions, I'd be glad to answer them. Thanks.
Thank you. Thank you very much, John, for that inspiring talk. And if you have questions from audience, you're free to ask now.
Oops. If I can go back to
Right, Mark. Mark, you're on mute.
A classic one. Hello, John. It's Mark here. John, as you said, neuroscience and neurosensing is an evolving field regarding this, as well as then, which I currently would say that you have right now also non-invasive neurosensing and invasive neurosensing regarding this. So, would you say that the profession needs to also research more in terms of grading a patient in the future and diagnosing that this patient is going to receive an invasive BCI and this patient needs to go towards non-invasive BCI therapy?
Well, I think that you know the issues. This points to this interdisciplinary culture that's necessary. The neuroscientist or the engineer might think of a solution and say, I'm going to develop that solution, but not think of its particular clinical application and the particular need of the patients. So if you think about the young woman I showed that's locked in, and you were able to communicate with her, you would say, what is the most important thing for you to have? For that person, the most important thing is to be able to communicate, be able to tell them that, my head hurts or I need it, I have a headache. For a person like a hemiparetic person that I showed you with the paralysis of the hand, the most important thing for them would be to pick up a pen and write or be able to type on their keyboard again. They can speak fine. They're relatively mobile. So we have to assess the needs of the patient. And then we can ask, where can we bring technology to bear to help each of those people? Now, going back to, say, a completely paralyzed, locked-in person, if you can't provide them the kind of speech that I showed you that you can do up to 90 characters per minute with an implantable device, maybe to start to convince the patient that it might be, that this technology can work, you would start with something very simple. And there are EEG-based devices that you can wear a cap and you can communicate with what's called a P300 device. For me, the problem with that is it's extremely slow and it's really tedious. But of course, if you have no way of communicating, that's a solution, and it's a rather straightforward one to use. And you can go out and buy a system now, today, and use it. But I think we can do much better. I think another misleading thing is that surgery is a serious event. I mean, we never, downplay when you do surgery to put a device inside your brain, and most people think their brain is a very precious structure, precious organ. So we take that very seriously. On the other hand, the complication rate in brain surgery is about the same as any other kind of surgery. And there's, unbelievable success in surgical implantation. We've published already safety data from the array that we've used. and that safety data is very promising. It looks very good. And so I think that, we will eventually have a range of solutions, and it'll be up to the patients to say which ones those are. And we need which ones are desirable to them. And we need to know, if people are saying, I think implantable devices are acceptable, or I think that wearable devices are the only thing that I want. And maybe just one last point. I think that if you look at the model of cardiac medicine, it's really a good one to follow. Because in cardiac medicine, you say, is there a cure for a cardiac disease? No, there's many. There's drugs, there's stents, there's vascular solution, there's valves, there's stimulators, there's implants, there's artificial hearts. So we have an enormous range of solutions. And the field, there isn't one brain technology versus another. We need a range of solutions because patients will have a range of needs and a range of wishes of what they would like to have. So that's, I think, it's a long and complex, a little bit of a complex answer, but it's still, I think, as I said, if we use the cardiac medicine example, I think it will show us the future of where we're going to go. And we don't really know which of these will be the most significant. I think they all have their value.
Thank you for your answer. And yes, you're correct, but you gave once a very good example. For example, when the pacemaker came out on the market, the first one, then the patients had a lot of resistance towards it. Now the market accepts it, and people say that why doesn't he or she have a pacemaker? So maybe there will be a small resistance at first, of course, and well, we need to be aware how we are going to build a way that reduce that resistance so that we can successfully introduce also invasive BCI systems.
Yeah, that's where the clinicians become important. The cardiologists face this. They the first cardiologist, I'm sure, when they explained we're going to stick wires in your heart and implant an electrical stimulator in your chest, that most people were saying there was no way I would have that done. But you have these pioneers like Matt Nagel and Kathy, and some of the other patients that you've met that are, want to be at the very frontier of doing these, participating in these kinds of evaluations. And then eventually there's acceptance. And this is the whole history of medicine is like this.
Thank you.
Are there any more questions?
I have a question, if possible.
Go ahead, Jacob.
Great talk, very fascinating. My question is about what is your idea to improve the performance of these type of restoration of function, if we think about hand, arm? Do you think that maybe what do you think about the sensory side and the integration of the sensory stimulation, like touch proprioception, into the loop? Do you think is a fundamental step for that, or?
Yeah. Oh, yes. I think, what we want to do is recreate all of the pathways that we normally use to work. Without sensory feedback, we have to use vision. We basically completely occupy our visual system to know what our hand is doing. So in order to recreate actual movement, and all engineers ask me that same question, why have you neglected touch? I don't. It's just we haven't worked on it. It's a very important problem. So it's tough. So we have two ways. Well, if in a spinal cord injury, the pathways are cut. So we have this problem of being able to put sensors in, say, in the hand and the fingertips and in the arm, and then get those signals into the brain. The same kind of electrode arrays can be used to stimulate. There are a number of groups that are working on showing how you can, in fact, recreate perceptions of touch and proprioception in the brain. The sensors are more complex, to get a sensor in your finger that can detect as your skin does, and then transmit that to something that's in your brain. But I'm confident that it can be done. But what is it? It's lots of sensors, very small, low power. I neglected to talk about all the huge engineering challenges that there are. But, these things are still need to be at a scale that are at the forefront of engineering. They need to consume power that is just at the very edge of what we're able to do. But in fact, industry is doing that. If you look at what's inside an iWatch or a iPhone or, these kinds of things, they're really extraordinary. And I actually think we have not used the full capabilities of what the semiconductor industry can do. So I think, these can be done. As you know, probably there's people who make gloves with artificial skin that have really remarkable properties, not as elegant as our skin, but some of the advances are very impressive. So I think this can be done. I think it's very important, and it's probably given a little bit less attention than the brain-computer interfaces for movement because the impact is so dramatic.
Yeah. John, if I could add to what you said, John. I mean, like in this project B-CRATOS, we are also having triboelectric nanogenerators for electronic skin, and which would be connected to the somatosensory cortex of the brain to give a feedback loop into the system. So this is also something that we are looking into in our project as well.
Yeah, I think the one thing that has really failed in that whole domain is that when you feel something, it's actually, again, it's back to those QR codes that I showed as a pattern of activity. And most of the efforts that I've seen are trying to recreate, say, touch on the fingertip with the shock of a single site in the brain. And I think that's a complete mistake because touching your finger gives rise to a very complex spatial temporal pattern. And we need to be able to recreate that pattern. And electrical stimulation does something that's not natural. It's very complex, and we need to understand that better. And I think that's a piece of the sensory reconstruction that's been neglected. I think people are coming around to it, but it's, it'll work with a single shock, just like I can alert you that somebody has touched your hand by tapping on your back, but it's not exactly the ideal way of what you want is to recreate touch that's as natural as possible. As I said, the goal should be eventually we have someone that's indistinguishable from you and me. If they want to go play basketball or soccer, they can go do it, and you wouldn't know that they had had a spinal cord injury or a stroke. That's an extraordinary vision. I mean, it's going to take a long time to do that, but I think it's achievable. Oops, I lost you, Robin.
Thank you, John. I was on mute.
Okay.
So it seems like we don't have more questions here. So, and we are already an hour into the talk. I would like to thank, on behalf of the audience, Professor John Donoghue for this hugely inspiring talk regarding all the possibilities in front of us with neuroelectronics, making people's life better and more
Maybe Robin,
qualitative. Thank you.
I'll end by saying, and I expect the B-CRATOS team to solve a lot of the problems I brought up. So it's on all of you.
And thank you, John, for putting us on track and being with us.
Good.
Thank you so much.
Okay.
And I hope to talk to you soon. And thank you all for attending this webinar. Thank you.
Thank you.